Welcome back. Today we are going to be discussing Chapter 5, Part 2, where we're going to discuss the synthesis, storage, and function of neurotransmitters. In our last class, we discussed the two key types of synapses, chemical synapses and electrical synapses. Remember that chemical synapses are used more often than electrical synapses, although we did cover both. Here's a quick refresher. Action potentials propagate down the axon of the presynaptic neuron, and when an action potential reaches the synapse, it's going to open voltage-gated calcium channels. When calcium comes into the presynaptic neuron, this initiates the release of neurotransmitters from synaptic vesicles. Now let's say we have a neurotransmitter that comes and binds to a receptor on the postsynaptic neuron. If this receptor is permeable to a particular type of ion, this neurotransmitter can initiate a conformational change, which will allow that ion from the extracellular fluid to enter the postsynaptic neuron. When ions enter the postsynaptic neuron, this can change its membrane potential. This could potentially depolarize the postsynaptic cell and initiate another action potential. But it can also alter gene expression or induce other biochemical cascades that we're going to talk about later today. Electrical synapses also utilize action potentials to generate electricity. When action potentials reach gap junction channels, ions can directly flow from the presynaptic cell's cytoplasm through the gap junction channel into the cytoplasm of the postsynaptic cell. Remember that gap junctions are made up of these tiny subunits of connexin proteins that form two connexons. This ultimately creates a channel, and these gap junction channels are in a very small space that's only about 3 nanometers in size. When ions come into the postsynaptic cell, they can generate postsynaptic potentials. And if you have multiple neurons that are sitting next to each other and they are firing these postsynaptic potentials, this coupling of postsynaptic potentials can generate depolarization. Another action potential that begins in this postsynaptic neuron can increase ions to flow back to the presynaptic neuron. This is why an electrical synapse has a bidirectional relationship. Ions can flow from the presynaptic neuron to the postsynaptic neuron, or ions can flow from the postsynaptic cell back to the presynaptic cell. Chemical synapses do not have a bidirectional relationship, and that's a defining quality of electrical synapses. Now remember that chemical synapses are actually more unreliable because they run the risk of becoming blocked. Electrical synapses have a far less risk of becoming blocked, and if they are, it can lead to detrimental consequences. But I do want you to remember that chemical synapses occur when neurotransmitters are released from the synaptic vesicles in the synaptic cleft, whereas electrical synapses are the direct transfer of ions from the presynaptic neuron to the postsynaptic neuron. Chemical synapses require ligand-gated ion channels and other types of receptors that we'll talk about later today. These ion channels, or receptors, sit on the postsynaptic neuron and allow the transfer of neurotransmitters from cell to cell. These neurotransmitters can also initiate the opening of ion channel gates, which will allow ions to enter the postsynaptic neuron. Electrical synapses require gap junction channels. Remember, this is a very small space that allows ions to transfer from the cytoplasm of the presynaptic cell to the postsynaptic. And finally, chemical synapses are not bidirectional, whereas electrical synapses are. And this is due to the generation of postsynaptic potentials. And we're going to talk more about those postsynaptic potentials later on in today's lecture. Remember that most synapses in the brain are chemical. Now, in my last lecture, we discussed two different types of synapses, including electrical synapses and chemical synapses. Electrical synapses are still important, although they occur less frequently. For example, we talked about how they play an imperative role during development. But most synapses in the brain are chemical, so thus we're going to focus on the different chemicals, also known as neurotransmitters, that are playing a role in synaptic transmission. Most neurotransmitters are either amino acids or modified amino acids, known as amines, or shorter strands of amino acids known as neuropeptides. different neurotransmitters are actually synthesized in different ways. So let's take a closer look. The three major categories of neurotransmitters are amino acids, amines, which are formed from amino acids, and peptides. Now we're largely going to focus on categories 1 and 2, but we will briefly discuss the formation of peptides as well. Here are the key neurotransmitters that we're going to focus on in class. So although there are 3 different types which I've talked about, including amino acids, amines, and peptides, which are not shown here, I created this table for y'all because these are the ones that I want you to really focus on. We're going to talk about how each of these amino acid neurotransmitters are synthesized and how all of these different types of amine neurotransmitters are synthesized. We're going to go through each one individually. I also uploaded a link to this handout so you can print it out or keep it on your computer to study. But these are the amines and amino acids that you will need to know for the first exam. So let's go through these step by step. First, we're going to discuss amino acids. First, we're going to talk about the neurotransmitters that are derived from amino acids. And we've talked about several different types of amino acids in class before, because remember that amino acids, when they come together, develop our first primary protein structure. So most synapses in the central nervous system are mediated by the amino acids GABA, glutamate, and glycine. And we're going to talk about all 3 of these individually. First, glutamate is the key excitatory neurotransmitter that's going to increase excitement. Glycine and GABA are typically inhibitory neurotransmitters, meaning they're likely to inhibit actions of the postsynaptic neuron. Now let's look at these more in depth. First, we know that glutamate and glycine are among the 20 amino acids that are the building blocks of protein. And we know that amino acids are developed during translation of messenger RNA to protein. Remember that the primary structure of protein is simply a single strand of amino acids that come together to form a polypeptide chain. Now, glutamate and glycine are abundant in all cells of the body, including neurons. But GABA is interesting because it's primarily made by neurons and primarily created by the neurons that release them. GABA works to inhibit neural signaling, similarly to glycine, but it's more common in neurons, whereas glutamate and glycine are abundant in all cells of the body, including neurons. So that's a key difference between those. Now, glutamate is one of the key excitatory neurotransmitters utilized in the brain. Glutamate helps us with our motor function, emotional processing, learning and memory, and neuroplasticity, which this may be a new term for you, but this is like synaptic plasticity. So essentially, this increases the amount of synapses that we can produce to increase learning and memory. We know that these synapses need to be consolidated for long-term memory storage, and we're going to talk about that in our later chapters in this class. Also, glutamate plays a really important role in information processing. We know that the main glutamate receptor in the brain is the NMDA receptor. It's both ligand and voltage gated, so we'll talk about how that works. And essentially, glutamate is absolutely necessary for learning and memory. GABA is an inhibitory neurotransmitter. It plays a role in reducing neural excitability, helps in the stress response, our sleep, and our muscle tension. While glycine is also inhibitory and plays a role in our motor coordination, cognition, vision, audition, and sleep. So as you can see, these neurotransmitters play many vital roles in the body, and we're going to discuss how they're created later on. Next, we're going to talk about a second type of neurotransmitter in the class known as amines. Now, amines are modified amino acids, and they are also small molecules, meaning they're carried in synaptic vesicles. Now, amines are simply modified versions of amino acids. And the amines we're going to focus on include acetylcholine, serotonin, dopamine, norepinephrine, and epinephrine. I also want to note that there's a subcategory of amines known as catecholamines. This includes dopamine, norepinephrine, and epinephrine. Catecholamines are all derived from tyrosine, and we're going to talk about how they develop from tyrosine. Additionally, I want you to know that serotonin is derived from tryptophan, and acetylcholine is derived from extracellular choline. And we'll talk about ways that you can make sure you have enough extracellular choline through diet. So the amines we'll focus on include acetylcholine, which we know is the very first neurotransmitter that was ever discovered by Otto Loewi, and it plays a role in our motor behavior, neuroplasticity, attention, and memory arousal. Based on Otto Loewi's experiment, we also know it plays a really important role in our heart rate. Dopamine is our pleasure neurotransmitter. It's released when we are doing something that makes us happy. It's involved in the reward system in the brain. It helps us with our planning, motivation, learning, and motor movements. Norepinephrine is also known as noradrenaline, and that is the British term for norepinephrine, noradrenaline, and it plays an important role in our mood, our arousal, our memory and stress response, our attention, our vigilance, maybe when we're coming into a stressful situation or an unsafe situation, and it even helps monitor our blood pressure. Epinephrine is also known as adrenaline, so you're probably very familiar with this term, but adrenaline plays an important role in our stress response, our heart rate, our blood pressure, and our fight or flight. We're going to discuss the fight or flight system, which is regulated by our sympathetic nervous system, which plays a really important role in activating heart rate, increasing our blood to the heart to make sure that we can get away from a stressful situation. Additionally, we're going to discuss serotonin, which is a neurotransmitter that makes us feel happy and can help improve our mood. It plays a role in our eating behavior, sleep, motivation, decision-making, learning and memory, and even vomiting. So, as you can see, these amines play a vital role in several different body responses as well. So, the amino acid and amine neurotransmitters are known as small molecules. They contain at least one nitrogen atom, and they're stored and released from synaptic vesicles. It's really important to note that small molecules are different from neuropeptides or peptides. Remember, that's the third type of neurotransmitter that we haven't gotten to yet. But these are small molecules, so they can be stored in synaptic vesicles, whereas peptides and neuropeptides are stored differently, and we're going to talk about that later on. The amine acetylcholine helps to mediate fast synaptic transmission at all the neuromuscular junctions. For example, we know that it plays an important role at the heart. It is made by extracellular choline. Extracellular choline is typically obtained through diet. So dietary choline is absolutely vital. If we don't get enough dietary choline, it's going to be difficult to synthesize enough acetylcholine in our nervous system. Interestingly, this is one of my areas of expertise. So I've looked at how choline can affect the brain, behavior, mood, and the best sources to get it from. Eggs are very high in choline, specifically the egg yolk itself. And we also know that cruciferous vegetables and leafy greens are high in choline. Many different types of fruits and vegetables, and beans, legumes, nuts. These are all very rich in choline. So make sure you're getting this in your diet. Now, dopamine, epinephrine, and norepinephrine are also amines, but they're also known as catecholamines. And this is because it is a compound that has a catechol and a side chain amine. Remember, we've talked about side chains, which are also known as R groups, when we were talking about how proteins are synthesized. If you remember when I was talking about side chains and R groups, this is when we were discussing the tertiary structure of proteins and how this particular compound can help a protein decide what shape it's going to take on. So for example, This side chain in particular is going to help define these proteins to become catecholamines. Catecholamines are all derived from tyrosine. Now, dopamine is referred to as the pleasure chemical in the brain because it's released when we anticipate or receive an award. This reward could be food, drugs, sex, etc. So, for example, if you're about to bite into a warm chocolate chip cookie, you're probably going to be producing dopamine. Or if you see a picture of a baby golden retriever puppy, you're probably going to be producing dopamine. So dopamine plays a role in many vital functions, aside from pleasure, including our learning and attention, our mood, our motor movements, our heart rate, kidney function, blood vessel function, sleep, pain processing, and even lactation. So let's talk about how dopamine is created. Dopamine is derived from the amino acid tyrosine. Tyrosine is converted to something known as L-DOPA. L-DOPA is the precursor for dopamine. So this is how it's created. So what is tyrosine exactly? Tyrosine is the precursor for L-DOPA and dopamine, and you can actually find it in plentiful food sources. Because I'm a nutritional neuroscientist, I'm really interested in seeing how different diets or different dietary factors can affect our mental health. And there are several mood disorders, such as generalized anxiety disorder or depression, that are related to decreased dopamine or serotonin levels. So thus, I think that we could try to incorporate diets that are rich in tyrosine or tryptophan, which are precursors of dopamine and serotonin, respectively. And that could potentially increase and help boost our mood. So I've been looking into some tyrosine-rich foods that maybe you could potentially add to your diet, and those include chicken and other types of poultry, dairy products like milk, cheese, and yogurt, avocados and bananas, even soy or tofu. So this is something that you could potentially incorporate into your diet, and maybe it could potentially help improve mood disorders. I'm really interested in seeing how this could be used in conjunction with therapy to potentially help patients with their symptoms. So remember that tryptophan is one of the 9 essential amino acids. And so serotonin is also known as 5-HT, which is a naturally occurring amino acid. Serotonin is our calming chemical, and it's known for helping with our mood. And so essentially, serotonin deficiency is highly linked to generalized anxiety disorder and depression. We know that serotonin plays other roles aside from our mood regulation, including appetite, sleep, learning and memory, and most recently, decision-making and behavior. So serotonin derives from the amino acid tryptophan. Tryptophan is converted into 5-HT. 5-HT is the precursor for serotonin. Interestingly, we know that the amino acid tryptophan can also be found in lots of different dietary factors. For example, serotonin can be found in fruits and vegetables like bananas or blueberries. It can be found in cocoa powder and different types of nuts, pumpkin seeds, walnuts, almonds, pistachios, sunflower seeds. So this is another way that I think we could potentially look at mitigating mood disorders, or to help at least relieve some symptoms of them, by trying to utilize new dietary patterns simultaneously with therapy. Now we're going to move on to neurotransmitters part 3. So I've already discussed how amino acids and amines are created. Now we're going to discuss peptides. This is a larger molecule, so it's different from the small molecule neurotransmitters, which are the amines and the amino acids. Peptides are larger molecules. They're chains of amino acids, and they're actually stored and released in something called secretory granules. So this is a little bit different from synaptic vesicles. Secretory granules, also called large dense core vesicles, function like storage compartments for secretory products within cells. So for example, here we're looking inside of the secretory vesicle containing insulin. Insulin is a vital hormone that's produced by the pancreas. You probably know that insulin resistance can play a role in diabetes mellitus type 2. And so thus, these secretory granules are in charge of keeping insulin inside for secretory products. Secretory granules and synaptic vesicles are frequently observed in the same axon terminals. So we know that they can also be stored at the synapse, and they are highly regulated in this area. Now, neuropeptides can also be short strands of amino acids, and they vary between 3 to 36 amino acids in length. These are large molecules. They're larger than the small molecule amino acids and amines that we discussed before. There are two examples that I want you to know. The key neuropeptides I'm going to talk about are oxytocin and vasopressin. Now remember, we've talked about them a little bit in my last lecture. Vasopressin is released to induce thirst, while oxytocin is our love hormone or bonding hormone. And these neuropeptides, which are essentially neurohormones, are produced in the pituitary. And so we are going to cover this more in chapter 15, but I just want it to be on your radar. So now we're going to discuss the synthesis and storage of different types of neurotransmitters. So there are two key ways that they can be produced. Peptide neurotransmitters are produced differently. And since we're talking about neuropeptides in these large molecules, I thought we would start with their production. So a precursor peptide is synthesized in the rough endoplasmic reticulum, shown here in purple. And then it's split in the Golgi apparatus, shown here in yellow, to yield the active neurotransmitter. Now remember that the rough endoplasmic reticulum is a key site of protein synthesis. So thus the proteins are produced here and then sent to the Golgi apparatus, which we've also talked about as kind of the post office of the cell, ensuring that peptides go where they need to. Secretory granules, as shown here in image 3, contain these larger molecules such as oxytocin or vasopressin. And secretory granules containing the peptide bud off from the Golgi apparatus, and they can be transported down the axon. Now remember, they're transported down the axon along microtubules, similarly to the way that synaptic vesicles are. They can be sent to the axon terminal, where that secretory granule will be stored. Now that we've discussed how peptide neurotransmitters are synthesized, we're going to focus on how small molecules, such as amine and amino acid neurotransmitters, are created. So once neurotransmitters are synthesized in the cytosol of the axon terminal, the amino acid and amine neurotransmitters must be taken up by the synaptic vesicles, as shown here in yellow. So let's use an example. We'll talk about serotonin here. We know the precursor molecule for serotonin is 5-HT, which is derived from the amino acid tryptophan. 5-HT will help develop the neurotransmitter molecule serotonin, and thus a transporter protein is going to need to move the synaptic vesicle down to the synapse. Concentrating these neurotransmitters inside the vesicle is the job of transporter proteins. These are special proteins that are embedded in the vesicle's membrane. So how do neurotransmitters get sent to the synaptic cleft? We know that this is known as exocytosis, and calcium is playing a key role here. Now, we've discussed equilibrium potential in depth before, but we've primarily focused on the equilibrium potential of sodium and potassium. The equilibrium potential of calcium is around 123 millivolts. Thus, there is very little calcium inside of the cell at rest. As you can see, at rest, we only have a few of these purple ions that are calcium. It has a strong driving force to go inside of the cell. Thus, calcium plays an important role in exocytosis. When an action potential reaches the axon's terminal, voltage-gated calcium channels need to open to allow calcium to rush in. So let's look at this step by step. Step 1, an action potential is going to arrive at the terminal. We know that once this action potential comes to the axon terminal, it's going to induce the opening of calcium ion channel gates. And calcium entry is going to cause neurotransmitters in these synaptic vesicles to be released. When neurotransmitters are released down into the synaptic cleft, this is playing an important role in synaptic transmission, specifically chemical synaptic transmission. Calcium's job is to tell synaptic proteins to get to work on the process of exocytosis. So a key takeaway I want you to know from this slide is that calcium must enter into the synapse to allow synaptic vesicles to release neurotransmitters. And this is known as exocytosis. Exocytosis occurs when the neurotransmitters are actually sent into the synaptic cleft. So this synaptic vesicle shown here is loaded with neurotransmitters and needs to release its contents into the active zone. Thus, these neurotransmitters can enter into the synaptic cleft, which is that small space in between the presynaptic neuron and the postsynaptic neuron. This all occurs due to the influx of calcium and wouldn't be possible without calcium. This is how small molecules, like amines and amino acids, are released from the presynaptic cell. Neuropeptides are larger in size, and because they're kept in secretory vesicles, their process of release is a bit different. I also want to mention something known as snare proteins. Snare proteins are important for vesicle loading and docking here, and they actually help to mediate exocytosis along with calcium. Now let's take a closer look at a chemical synapse. Here we have the presynaptic neuron and the postsynaptic neuron. So we would need an action potential, as shown here, to generate this nerve impulse that will go down to the axon terminal. Here, the action potential induces the opening of calcium ion channels. Calcium is going to rush into the presynaptic cell, and then it's going to increase the release of these tiny neurotransmitters from the synaptic vesicles. When neurotransmitters are released from the synaptic vesicles, they enter the synaptic cleft, where they can be taken up by receptors on the postsynaptic neuron. Remember, receptors are usually specialized and ligand gated, such that a certain type of neurotransmitter will induce its opening or binding. As you can see towards the end of this video, the action potential was generating in the postsynaptic neuron due to neurotransmitters coming into that cell. So thus, this is a cycle in which action potentials can generate these nerve impulses to increase the release of neurotransmitters to the postsynaptic cell. And when these neurotransmitters reach the postsynaptic cell, this can induce another action potential if depolarization occurs. Exocytosis can occur very rapidly, within about 0.2 milliseconds of calcium influx at the end of the axon terminal. And synapses in mammals, which generally occur at higher temperatures, are even faster. So how do neurotransmitters leave the synaptic cleft after they're released? There are several different ways that neurotransmitters can be removed from the synaptic cleft. First, we know that diffusion plays a role here, and we've already talked about passive transport versus active transport. We know that the sodium-potassium pump utilizes active transport to transport ions against their concentration gradient. And remember, that pump is helping to stabilize the amount of sodium and potassium inside and outside of the neuron. So we know that that pump is playing a role here. We also know that reuptake can help remove neurotransmitters. So we've already talked about these specialized transporter proteins. Transporter proteins can sit here at the membrane at the synaptic cleft and help take up extra neurotransmitters into the cell. So for example, these green neurotransmitters can be taken back up by this red transporter protein to go back inside of the cell. And we know that vesicular proteins help with endocytosis. So endocytosis is the process in which these neurotransmitters are put back into vesicles. Thus, this is opposite of exocytosis, in which neurotransmitters are actually released by synaptic vesicles. So here we're looking at reuptake of neurotransmitters in green and how transporter proteins basically take these neurotransmitters from the cleft and put them back into synaptic vesicles during the process known as endocytosis. We also know that they can be broken down by enzymes. So excess neurotransmitters that are left in the cleft can be broken down by different enzymes, such as monoamine oxidase. If you've heard of monoamine oxidase, this is actually a target of a drug known as monoamine oxidase inhibitors. This drug is also used to treat mood disorders, depression, and anxiety. So interestingly, monoamine oxidase is an enzyme that can break down important neurotransmitters that we need, such as serotonin. So this can be prescribed for the mood disorder, generalized anxiety disorder, to prevent excess enzymatic breakdown of monoamine oxidase, thus allowing more serotonin to remain in the synaptic cleft. Additionally, glial cells play a key role in removing excess neurotransmitters from the synaptic cleft. If you remember earlier in this class when we talked about astrocytic end feet, they do something called chemical buffering. And a glial cell's chemical buffering abilities allows the astrocytes around neurons to take up any excess neurotransmitters. In addition to monoamine oxidase inhibitors, selective serotonin reuptake inhibitors, also known as SSRIs, are one of the most highly prescribed medications for mood disorders. Now, SSRIs can be used for treatment of depression and generalized anxiety disorder because they influence serotonin levels. So now let's take a look at how SSRIs work. So essentially, SSRIs help to inhibit the reuptake of extra serotonin that's in the synaptic cleft. We know that transporter proteins, these red proteins here, can play a role in reuptaking neurotransmitters in the synaptic cleft in the process known as endocytosis. Essentially, SSRIs come and bind to transporter proteins and prevent the reuptake of serotonin, thus leaving more serotonin in the synaptic cleft. This is a really important mechanism for people who maybe produce lower levels of serotonin and thus suffer from generalized anxiety disorder or depression. So, do neurons only produce and secrete one type of neurotransmitter? The answer is no. Most neurons have the ability to produce and secrete multiple different types of neurotransmitters, such as small molecules and large molecules. So now that we've talked more about neurotransmitters, it's also important to know that certain neurons can be specialized to secrete specific type of neurotransmitters. However, I do want to note that neurons can typically produce and secrete multiple types of neurotransmitters. This is known as Dale's principle. So Sir Henry Dale was a British pharmacologist, and he described cells that produce and release acetylcholine as cholinergic cells. So thus, cells that produce and release serotonin could be referred to as serotonergic cells. And so essentially, he aimed to answer the question, do neurons only release one kind of neurotransmitter? And his principle proposed that a given neuron only releases one kind of neurotransmitter. However, this is false, because although there are some neurons that do produce high levels of choline or serotonin, they are also playing a role in the secretion of other types of neurotransmitters, typically. So some neurons only release one kind of amino acid neurotransmitter. However, others can co-release various neuropeptides like oxytocin and vasopressin, in addition to amino acid neurotransmitters such as acetylcholine. Now that we've talked about action potentials in depth, we're also going to talk about graded potentials. If you remember, we did discuss graded potentials in chapter 4 briefly when I talked about how these are smaller potentials. And a great example of graded potentials is the sodium-potassium pump. So now we're going to talk more about synaptic integration. And graded potentials are changes in membrane potential in response to channels opening. They generate something known as excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And we're going to talk about these in more depth. First, let's talk about excitatory postsynaptic potentials. I'll refer to these as EPSPs. So let's use our same example of a presynaptic neuron synapsing onto a postsynaptic neuron. The presynaptic neuron releases a neurotransmitter that binds to the postsynaptic neuron's membrane. So in this example, we could say that this neurotransmitter is glutamate. Let's say that glutamate is released and it is taken up by the postsynaptic neuron's membrane. A cation, like sodium, is going to bind to the ion channel, and the membrane will thus become depolarized in the postsynaptic neuron. Because sodium channels are going to open up and allow intracellular sodium to enter into the cytosol of the postsynaptic neuron, this is going to generate electricity. And this is called an excitatory postsynaptic potential, because the release of the neurotransmitter glutamate in the presynaptic neuron is triggering an action potential in the postsynaptic neuron. For example, a receptor on the postsynaptic neuron could be the NMDA receptor. An activation of that receptor due to the binding of glutamate could increase extracellular sodium to enter these channels and enter the cytosol of the postsynaptic neuron, thus causing depolarization, which is the first step of the action potential. We know that acetylcholine-gated and glutamated-gated ion channels generate EPSPs. Now let's talk about the inhibitory postsynaptic potential, or an IPSP. This is going to increase inhibition of activity in the postsynaptic neuron. So in this example, we have two neurons. We have our presynaptic neuron and our postsynaptic neuron. The presynaptic neuron releases a neurotransmitter that binds to the postsynaptic neuron's membrane. For this example, we could say that GABA, the inhibitory neurotransmitter, which is an amino acid, is released from the presynaptic cell and taken up by the postsynaptic cell. An anion binds to the ion channel, and the membrane is thus hyperpolarized in response to this postsynaptic neuron. In this case, our anion, or negatively charged atom, is chloride. This is called an inhibitory postsynaptic potential that I'll refer to as an IPSP, because the release of the neurotransmitter, and in this case I talked about it being GABA, the release of GABA in the presynaptic neuron is going to make the cell more negative in the postsynaptic neuron, thus generating this IPSP that's inhibitory. This inhibits the postsynaptic cell from triggering an action potential because it's going to cause the inside of the neuron to become more negative from the introduction of extracellular chloride inside of its cytosol. And we know based on our knowledge of membrane potential that if the inside of the neuron becomes more negative, that cell is at rest. So thus GABA is going to increase negative charge inside of the cytosol, causing the cell to become hyperpolarized, which means that the cell is even more negative than it is at resting membrane potential, thus preventing an action potential from occurring. So you can see how this is the opposite of an EPSP, in which sodium will enter the cell to cause it to depolarize and spike an action potential. This is a very opposite contrast because chloride is going to make it more negative, which is thus in turn going to help the cell rest. This is why GABA is the key neurotransmitter that plays a role in inhibition. And synaptic activation of either glycine-gated or GABA-gated ion channels can cause an IPSP. Remember that glycine is common in most cells, but GABA is mainly secreted and produced by neurons. Now let's recap some of the key topics we've talked about today. First, we know there are two different types of synapses, electrical synapses and chemical synapses. We know that electrical synapses are the direct transfer of ions from the presynaptic neuron to the postsynaptic neuron. We also know that these synapses are bidirectional, in which large amounts of postsynaptic potentials can trigger another action potential that will run from the postsynaptic neuron back to the presynaptic neuron. Now we're going to focus primarily on chemical synapses, as chemical synapses are the most commonly used synapses in the body. Chemical synapses require neurotransmitters, and we've talked about 3 key subtypes, including amino acids, amines, and peptides. Now that we have learned about these 3 classes of neurotransmitters, we're going to talk about 2 different key receptors. We've already talked about the neurotransmitter receptor, which is known as ligand-gated ion channels. Ligand-gated ion channels are also called transmitter-gated ion channels, or ionotropic receptors. And that is because when a neurotransmitter binds to these ion channels, it can open the gates if it's selectively permeable to a particular ion and allow that ion to enter the postsynaptic cell. So that is our first subtype of neurotransmitter receptor. And the second key type of neurotransmitter receptor we're going to be talking about are G protein coupled receptors. These are also known as metabotropic receptors because they can actually have an effect on the cell's metabolism. And just a reminder, these neurotransmitter receptors are typically sitting right here at the postsynaptic cell membrane. Now, at the postsynaptic cell, we need ion channels or receptors to take up any of this neurotransmitter that's being sent from the presynaptic cell to the postsynaptic cell. And both transmitter-gated ion channels and G protein-coupled receptors are the 2 key types of receptors that are utilized. There are several different types of neurotransmitter receptors, but these are the 2 key types we're going to focus on in this class. And each type has subtypes of receptors that are utilized based on the neurotransmitter at play. Based on what we've learned about chemical synaptic transmission, we know that neurotransmitters that are released by synaptic vesicles in the presynaptic neuron can enter the synaptic cleft, or the gap in between the presynaptic neuron and the postsynaptic neuron. We know that these neurotransmitters can come and bind to ligand-gated ion channels, or receptors, on the postsynaptic neuron. So for example, these receptors shown in this burgundy color can have a specific ligand or neurotransmitter that binds to it that induces conformational change. This conformational change simply changes the shape of the receptor and allows its gates to open. You can think of the binding of neurotransmitters to particular receptors like inserting a key in a lock. The neurotransmitters are like a key, and the receptor itself is a lock, and it can only be unlocked by certain ligands or neurotransmitters that will allow that conformational change to occur, thus allowing ions to flow from the presynaptic cell to the postsynaptic cell. Now, it's interesting to note that there are over 100 different neurotransmitter receptors, but they can be classified into 2 subtypes. One is transmitter-gated ion channels and G protein coupled receptors. Now let's talk about these 2 more in depth. So there are 2 major neurotransmitter receptor subtypes. We've talked a lot about ligand-gated ion channels, which are also known as transmitter-gated ion channels, because a neurotransmitter or a ligand needs to bind to that channel to induce conformational change, in which the channel will open its gates and allow ions to come into the channel and go into the postsynaptic neuron. This is why they're also known as ionotropic receptors, because it allows ions to pass through when a neurotransmitter binds to it. There's also another type of receptor known as a G protein coupled receptor. This is a metabotropic receptor. So what does this mean exactly? First, I want to clarify that G proteins are also known as guanine nucleotide binding proteins. That's a mouthful, so we're going to call them G protein coupled receptors. These receptors are not ion channels like the previously discussed receptors. They're metabotropic receptors because when a ligand or neurotransmitter binds to this receptor, it actually initiates metabolic activity inside of the cell and ultimately modulates the activity of the neuron. So we're going to talk about these 2 receptors in more depth. So first I'm going to discuss the ligand-gated, also known as the transmitter-gated ion channel. This channel is composed of multiple subunits of tertiary protein structures. So if you remember, these are 3D structures that come together to form one large structure known as the quaternary structure. This goes back to our lecture on the simple functions of proteins. So essentially, when these protein subunits come together and form the receptor, they form a pore in between them. And typically when the cell is at rest or that neurotransmitter is not present, the pore is going to be closed. So when neurotransmitters actually come and bind to specific sites on this extracellular region, it induces conformational change, which I've talked about many times before, but this is simply when the structure changes shape. And this occurs within microseconds and causes the pore to open. So the functional consequence of this depends on which ions can pass through the pore. And so in our example, we're going to talk about acetylcholine-gated ion channels. So let's go in more depth about the acetylcholine-gated ion channel. So for example, let's say that this transmitter-gated ion channel is selectively permeable to sodium. So if acetylcholine comes and binds to this channel, and it induces conformational change in which that channel can open up and allow ions to come inside of the postsynaptic cell cytoplasm, in this case, this channel is selectively permeable to sodium. So extracellular sodium could come inside of the channel and enter the cytoplasm of the postsynaptic cell. This would cause depolarization because, as we know based on our knowledge of action potentials, extracellular sodium that comes inside of the cytosol is going to depolarize the cell and make it more positive than it would typically be in its resting membrane state. Thus, because it's going to generate an action potential and depolarize the cell, this would be known as an excitatory effect. It's going to bring the membrane potential towards threshold for generating an action potential at around -55 millivolts. Now remember earlier I talked about how receptors basically are doors that have their own keys. So you can think of a ligand or a neurotransmitter as the key that needs to come and bind to this blue channel in order for ions to pass through into the postsynaptic neuron. These receptors will only open when that ligand binds to it. Normally, if there are no ligands or neurotransmitters present, it is going to remain closed. The pore and the channel only open up due to conformational change when a ligand binds to it. And again, ligand can be used synonymously with the term neurotransmitter. So in this example, sodium could come into the cell and potassium could leave the cell. This ion channel helps the membrane get closer to its equilibrium potential of sodium, thus increasing depolarization and the generation of action potentials. So when the cell becomes more positive, we know it overpowers the potential of potassium leaving the cell, and another action potential can occur. Remember that this is a cycle in which sodium can come into the cell and potassium can leave the cell. This is also regulated by sodium-potassium pumps that sit within the phospholipid membrane. All of these channels and pumps are working simultaneously to ensure that the cell has enough time to rest and then also to spike these nerve impulses known as action potentials. Now that we understand transmitter-gated ion channels, I want you to understand this key takeaway. Remember that transmitter-gated ion channels, also known as ligand-gated ion channels, are typically utilized for fast chemical synaptic transmission by different neurotransmitters, but these are the two small molecule neurotransmitters, the amino acid and amine neurotransmitters. So for example, this would include our catecholamines like dopamine or norepinephrine, or monoamines like serotonin. There is an additional type of receptor, and this is known as G protein coupled receptors, that actually have slower and longer lasting postsynaptic actions. All three types of neurotransmitters, including amino acids, amines, and the larger molecules, which are peptides, can act on G protein coupled receptors. So this is very pertinent for long-term learning, these slower, longer postsynaptic actions. But we'll get to that in a minute. Before we talk about their actual role, I want you to understand what a G protein coupled receptor is. So G protein coupled receptors are actually the largest known class of membrane receptors in the body. There are thousands of different types, and each type has a specific function. For example, 30 to 50% of all modern medicinal drugs target different types of G protein coupled receptors. They can help regulate the immune system and our mood via modulation of serotonin and dopamine, and even more. Hormones, pheromones, and neurotransmitters can bind to G protein coupled receptors. So, how are G protein coupled receptors different from ligand gated receptors? I'm going to use this video from 2 Minute Neuroscience, which you can utilize this link to look at the video yourself, in order to show you how G protein coupled receptors work. Here's a G protein coupled receptor in pink. We already know that the G protein coupled receptor is made up of multiple subunits, including the alpha, beta, and gamma subunits. These subunits come together to form this structure that's anchored to the phospholipid membrane by lipid anchors. When a neurotransmitter, such as this green neurotransmitter here, comes and binds to the G protein, it induces conformational change. The subunits of the G protein split apart. But for the scope of this class, we're not going to go into too much depth about that process. What I do want you to know is that when a neurotransmitter binds and induces this conformational change, it can cause other changes at the phospholipid membrane. For example, the G protein can influence the opening of ion channels nearby. And it can also affect other enzymes, such as second messengers. Second messengers help modulate other signaling cascades in the neuron and can actually influence the cell's metabolism. So let's walk through this together. As I noted previously, G proteins are made up of 3 subunits, including the alpha, beta, and gamma subunits. Here is our G protein shown here in blue. The G protein is anchored into the phospholipid membrane by these lipid anchors. So when a neurotransmitter, such as this red one here, comes and binds to the G protein receptor, it's going to induce conformational change, in which this protein is going to change its shape. The receptor proteins then activate these small G proteins, which are free to move along the postsynaptic membrane. The activated G proteins can function in different ways. First, they can function as effector proteins themselves. So for example, here's a G protein gated ion channel, and the conformational change of this G protein can allow this channel to open and allow ions to enter into the postsynaptic cell. But they can also be influenced as second messengers. G proteins can initiate the second messenger cascade, which can induce biological changes. For example, it can affect cellular energy and metabolism. Let's review this to look at the big picture. So when a neurotransmitter binds to this receptor of the G protein, we already know that it's going to induce a change in shape, known as conformational change. G proteins are composed of these 3 subunits, including alpha, beta, and gamma subunits. G proteins can influence the opening of nearby ion channels. They can influence other enzymes. And these enzymes can activate signaling molecules that are known as second messengers. Second messengers essentially induce a signaling cascade. So they can increase more complex intracellular signaling, and this process takes much longer and it's slower than ligand-gated ion channels. Because this process takes more time and it's slower, it can induce more permanent changes. That's why G protein coupled receptors are actually super important for the neural mechanisms of learning and memory, because it's going to induce longer lasting changes, such as taking the input of a memory and consolidating it for long-term storage. these effects induce long-term postsynaptic change. And for example, it can induce transcription and translation and thus affect gene expression. So second messengers have the ability to induce transcription and translation of new proteins. And we know that the final impact of gene expression is from protein synthesis. So genes can be expressed due to this initiation from second messengers. We can use an example such as transcription of a protein known as brain-derived neurotrophic factor. Second messengers can initiate the transcription and translation of the BDNF gene. BDNF plays a vital role in learning and memory. This neurotrophic factor is actually released when we work out, and newer research has even shown that BDNF is released when we eat healthy food. Since BDNF is really important for supporting our cognition, it's important to understand how G protein coupled receptors can actually initiate this cascade for transcription and translation, and thus gene expression. So essentially, my big takeaway from this slide is that G protein coupled receptors can initiate multiple different types of cascades, including the opening of nearby ion channels or influencing enzymes that can initiate this cascade that could potentially lead to long-term postsynaptic change. And that's why this receptor is important in learning and memory. So, how do drugs act on the neuron's receptors? There are several drugs that have been created to help with mood disorders and neurodegenerative disease. Many of these drugs are created as antagonists to block enzymatic breakdown of specific neurotransmitters that are involved in those diseases. Let's start with the example of Alzheimer's disease. So, cholinesterase inhibitors have been utilized as a drug for Alzheimer's disease. However, I do want to note that cholinesterase inhibitors do not slow down the actual progression of the disease. They only help with some of the symptoms like memory loss in early stages of the disease. However, it's not a long-term treatment, and sadly, Alzheimer's disease does not have a cure. We have not been able to find anything that can actually reverse the effects of Alzheimer's, as it is a fatal disease. But in earlier stages, cholinesterase inhibitors, such as the drug Aricept, have been really helpful in mitigating those cognitive deficits and cognitive impairment that patients experience. And so cholinesterases are a class of enzymes. Remember, enzymes typically end in ACE, A-S-E. So cholinesterase is an enzyme that is produced in the body to break down choline. And obviously, we understand that acetylcholine plays a really important role at sites like the neuromuscular junction, but cholinergic neurons in the brain also help with learning and memory. In the earlier stages of Alzheimer's disease, there's a large deficit of cholinergic neurons in the basal forebrain, and the axons of those neurons project into the hippocampus. We know the hippocampus is vital for learning and memory, as it's one of the key areas where learning and memory is processed and consolidated. So thus, cholinesterase inhibitors can prevent that enzymatic breakdown. They inhibit those enzymes, and thus they can be used as antagonists. There are other drugs that are also used as antagonists or inhibitors for other types of mood disorders. We've already discussed monoamine oxidase inhibitors. And these are drugs that actually block the reuptake of monoamines. And monoamine oxidase is a specific type of enzyme that breaks down monoamines such as serotonin. These drugs are utilized to help people with generalized anxiety disorder or depression. And finally, we've also talked about selective serotonin reuptake inhibitors and how they inhibit the reuptake of serotonin out of the synaptic cleft. And they particularly prevent transporter proteins from performing endocytosis, which is the reuptake of extra serotonin in the synaptic cleft. And so thus SSRIs have been helpful in mitigating symptoms of anxiety, depression, and other mood disorders. So thus you can see why it's important to understand how antagonists work, as many drugs in the field of psychology and psychiatry are utilized to treat these disorders. Now we're going to discuss agonists versus antagonists. So most neurotransmitters are named after their agonists. So interestingly, an agonist is a chemical or drug that actually binds to the receptor and mimics the actions of the naturally occurring neurotransmitter. So for example, an agonist of dopamine would simply mimic dopamine, and it would bind to the receptor and mimic the response of dopamine in the neuron. These drugs are known as receptor agonists. They can be excitatory or inhibitory depending on the type of neurotransmitter they're trying to mimic. For example, if they were mimicking glutamate, they could be excitatory. If they're mimicking the actions of GABA, they'll likely to be inhibitory. Antagonists are different from agonists. Rather than mimicking the neurotransmitter, it actually blocks the neurotransmitter from coming into the receptor. So these are inhibitors of neurotransmitter receptors, and they bind to the receptors and block or antagonize the normal action of the neurotransmitter. So it simply sits on the receptor and blocks it. Here's what this looks like. So let's first focus on agonists. So as you can see, an agonist is a type of chemical or molecule that will come and actually bind to the receptor and mimic the action of the naturally occurring neurotransmitter. Whereas an antagonist actually comes and blocks the receptor's gate. It won't allow anything to come into its channel, and it produces no effect. It's not trying to mimic the neurotransmitter that's naturally occurring. These are two common types of drugs. So let's look at some examples. For clarification, let's look at two different types of agonists. First, we're going to talk about nicotine that comes from tobacco. And second, we're going to talk about muscarine. This is an agonist that actually is utilized as a drug treatment for Alzheimer's disease, glaucoma, and irritable bowel syndrome. So nicotine is an agonist at the nicotinic acetylcholinergic receptor. And I know that's a mouthful, but this is a particular type of acetylcholinergic receptor for nicotine. When someone ingests nicotine, it actually stimulates this receptor. And I drew this picture as a reference so we can walk through each step. Nicotine can come and bind to this ligand-gated acetylcholinergic receptor in skeletal muscle. And so nicotine is an actual agonist for this. And if you remember, an agonist mimics the naturally occurring neurotransmitter. This happens at the neuromuscular junction, so all of the neurons that are typically synapsing onto muscle. This is also an ionotropic receptor. So when nicotine comes and binds to this ligand-gated ion channel, it will induce conformational change, in which this channel can now open. This will allow extracellular sodium to come inside of the neuron, whereas there could be an efflux of potassium. And we know during this first step, when extracellular sodium comes inside of the cell cytosol, it's going to induce depolarization, which, of course, can stimulate an action potential. There are also nicotinic acetylcholine receptors present in our central nervous system, and these are involved in the addictive behavior with tobacco use. I also want to note that this receptor generates an excitatory response, hence the reason I put this plus sign here. So remember that nicotine is an agonist, and thus it's mimicking the naturally occurring neurotransmitter and stimulating an excitatory response. And it's generating excitation by allowing extracellular sodium to come into the cytosol and depolarize the cell. Muscarine is an agonist at the muscarinic acetylcholinergic receptor. So this is similar to nicotine, except it works at the G protein coupled receptor instead. Muscarinic agonists activate this particular receptor and help aid in digestion, heart rate regulation, and even memory formation. So muscarinic agonists have been used to treat glaucoma, Alzheimer's disease, and irritable bowel syndrome. This is a G protein coupled receptor, and it is inhibitory, meaning it's going to stop activity. So let's take a look at this in more detail. So the muscarinic acetylcholinergic receptor is a G protein coupled receptor. It plays a really important role at our heart muscle to help regulate our heart rate. So here's an example. We know that G protein coupled receptors are made up of these 3 different subunits, including the alpha, beta, and gamma subunits. We know that this protein structure is anchored to the phospholipid membrane with these lipid anchors. So based on what we know about G protein coupled receptors, we know that when a neurotransmitter comes and binds this receptor, it can influence nearby ion channels. So when muscarine comes and binds to the G protein, it initiates this cascade in which nearby ion channels can open up. Specifically, when muscarine comes and binds to the G protein, it's going to induce an efflux of potassium. So potassium will be released from the cell into the extracellular fluid. Based on what we know about action potentials, we know that this efflux of potassium is going to generate hyperpolarization and inhibition of activity, which will slow down our heart rate. This is a parasympathetic response, and the parasympathetic nervous system plays an important role in resting and digesting. We're going to talk more about these different parts of the nervous system in our next lecture. But this goes back to Otto Loewi's experiment. Remember when I described Otto Loewi's discovery of Vagostoff, which is known as acetylcholine now, and that was the very first neurotransmitter that was ever discovered. So essentially, in his experiment, when the heartbeat slowed down in the first frog heart and then transferred to the solution to the second frog heart, we know that acetylcholine was being released, and it was helping to slow down the heart rate. Now we know that acetylcholine works at this muscarinic acetylcholinergic receptor in order to help slow down the heart. So essentially, the binding of muscarine to this G protein coupled receptor helps change the excitability of the neuron. Now that we've talked about 2 different types of agonists, which, if you remember, agonists come and bind to a receptor and mimic the naturally occurring neurotransmitter, antagonists work a little bit differently. Antagonists can actually block the receptor and inhibit activity. A great example of a receptor antagonist is curare. This is a poison that's found in South America, and South American natives actually coat their arrow tips in curare to paralyze their prey. If you've heard of the show The Green Arrow, and if you're a fan of The Green Arrow, you probably know that Oliver Queen has come in contact with curare before when he was stranded on an island. This is a very dangerous toxin. Curare binds tightly to acetylcholinergic receptors on skeletal muscle cells and actually inhibits the actions of acetylcholine. By inhibiting acetylcholine, it prevents muscle contraction and thus induces paralysis in its victims. There are lots of different poisons that can act on the neuromuscular junction. We're going to talk about botulinum toxin, famously known as Botox, black widow spider venom, and cobra venom, and how they affect neurotransmitter release at the neuromuscular junction. Botulinum toxin actually comes from a bacteria. It's a neurotoxin that works at the neuromuscular junction. Interestingly, most people don't know when they are using Botox injections that they are literally injecting a neurotoxin into their face or other areas of their body. Since muscle contractions can actually cause wrinkles, if you inactivate those muscles, you can smooth the skin and look younger. That's how Botox works. It's actually neither an agonist or antagonist because it works presynaptically. So what does this mean exactly? It essentially messes up with snare proteins. If you remember, I briefly talked about snare proteins and the role they play during exocytosis. Snare proteins are involved in helping synaptic vesicles full of neurotransmitters fuse with the presynaptic membrane in the terminal. And if these proteins are disrupted, there is no vesicle fusion. Thus, the activity between the neuron and muscle can't occur, it's inhibited, and that can prevent wrinkles. So this is how Botox works. Botulism is caused by several kinds of botulinum neurotoxins, and they're produced by the growth of C. botulinum in improperly canned foods. You may have heard of instances like this where people have become infected. The name itself comes from the Latin word for sausage because it was usually associated with poorly preserved meat, and this neurotoxin could be highly dangerous to ingest. Now we know that botulinum toxins are utilized as very potent blockers at the neuromuscular junction, and they inhibit neuromuscular transmission. So it's estimated that as few as 10 molecules of the toxins are enough to inhibit a cholinergic synapse. Now that we know much about cholinergic synapses, we could see how botulinum neurotoxins could be very dangerous if they are not handled properly, such in the case of poorly preserved meat, although they are used commonly for cosmetics today. Another example of a poison or venom working presynaptically is black widow spider venom. Sadly, we do have black widow spiders here in California. I've even seen them at my parents' house, so beware of them. They also act presynaptically like botulinum toxin, but they function in a slightly different way. So black widow spider venom actually initiates the release of a bunch of calcium into the neuromuscular junction. We know this as exocytosis. So when there's plentiful calcium that's abundant in the terminal of the presynaptic cell, it causes that area to swell. And all of the synaptic vesicles carrying neurotransmitters are then dumped into the synaptic cleft. This huge release of neurotransmitters actually causes the synapse to become inactivated. And this is why you can actually die from a black widow spider bite. Cobra venom is different and functions differently from the two previously described poisons. Like curare, it is an antagonist, which means it blocks the receptor. And cobra venom specifically blocks the nicotinic acetylcholinergic receptor. So it prevents acetylcholine from doing its job at the neuromuscular junction. Interestingly, the bite of a Taiwanese cobra also results in the blockade of neuromuscular transmission in its victim with another mechanism. One of the compounds in this particular snake's venom is called alpha bungarotoxin. This is a peptide that actually binds so tightly to the postsynaptic nicotinic acetylcholinergic receptors that it takes days to be removed. This bite can be fatal if not treated quickly. Usually there's no time for removal. However, because the toxin prevents the activation of nicotinic receptors by acetylcholine and paralyzes the respiratory muscles of the victims, this is probably one of the most potent poisons that you could come across.